In this lecture, we're going to take a look at where, why, or how the body will go out of acid-base balance, and then one of the first steps that we have for restoring homeostasis around pH in the body, and that's the chemical buffer systems that are found in our body solutions. So before we get started, we need to define a couple of terms, acids, bases, and I want to just do a quick review for you about pH. So recall that pH is a scale that measures how acidic or basic a solution is. It's a scale that goes from 0 to 14. So some hellish evil demon came up with this crazy scale. And right at the middle, at pH 7, is what we think of as neutral. And as hydrogen ions accumulate, then pH becomes lower and lower and lower until we get to the most acidic possible, which would be 0. So we get to a smaller number as we increase the concentration of hydrogen ions. So acids are materials that drop off hydrogen ions into solution when you dissolve them into a water in water. And so acids cause pH to decrease. If, on the other hand, we dissolve into the water a basic substance, it will release substances that, release compounds that will combine with hydrogen ions, or that material will actually mop up those hydrogen ions and remove them from solution, so that we get fewer and fewer hydrogen ions in the solution. And when that occurs, then we see an increase on the pH scale toward the more basic end of things, sometimes talked about as alkaline. So basic and alkaline, they mean the same things. So if hydrochloric acid, HCl, when you dissolve that in water, so HCl in water, water will get in there and interfere with the bond between the hydrogen and the chlorine atoms. And when they break apart, hydrogen donates one of its electrons to chlorine, and that makes the chloride ion out of chlorine. And the hydrogen, which has now lost its electron, has a positive charge. So there's our source of acid when we dissolve hydrochloric acid in water. It's the hydrogen ion electrolyte that's produced. Now, if we put a base into solution, then we can think of something like sodium hydroxide is a base. When we put that into water, it also ionizes, and we wind up with a sodium ion because the sodium gives away its electron, and it gives it to an OH minus. So how in the world does this OH minus remove hydrogen ions? Well, let's run a quick little chemical reaction here. If we take the hydroxide ion, that's an OH minus, and we react it with a hydrogen ion, what do we get? Hopefully you realize that you get OH and an H, which is, oh look, it's the same thing as H2O. So when you react a hydroxide ion with water, with a hydrogen ion, you wind up with water as your product. Very cool. As well, sodium bicarbonate. Now let's think about Grandma. Back in the olden days, if Grandma had a sour stomach, well, she'd just go into the kitchen and she'd mix herself up a glass of water with, hopefully you've now said, baking soda mixed into it. Baking soda is sodium bicarbonate. So when you dissolve sodium bicarbonate in water, it releases a sodium ion. We would have guessed that. And that sodium ion, it's positive because it gave its electron to the bicarbonate ion. You now have, well, the reason Grandma has a sour stomach is because there's a lot of acid going on in there and it's burning her up. So the acid, which is hydrochloric acid, by the way, those hydrogen ions that are in her stomach then will do a chemical reaction with this bicarbonate ion, producing H2CO3. Hopefully you recognize that as, I'll give you a chance to think about it, carbonic acid is what you should have said. If you didn't, ooh, shame on you. Okay, not really. But I am going to ask you to learn these chemical reactions this time. This chemical reaction, especially the one that we're seeing right here, is going to be an important one for you to be aware of. And it's a reversible reaction. It can go either way in our bodies when we do this. Now, the reason that Grandma gets all belchy after she's drunk her sodium bicarb is because, hopefully you remember this as well, that carbonic acid will fall apart into water, H2O, and carbon dioxide gas, and that's what Grandma's belching out, is that carbon dioxide gas. So there are many other basic chemicals in the body and in nature. Let's see, ammonia, NH3, is very basic. I think it sits somewhere out here at like 11 on the pH scale. If you combine ammonia and hydrogen ions, they will chemically react and form NH4+. Boy, I'm making a big old mess of this slide. Sorry about that. Hopefully you've been following along and you can figure out what I'm saying. So ammonia can even help to neutralize the hydrogen ions. Now, there's hydrogen in this ammonium ion here that we're looking at. But because it's not a hydrogen ion just loose running around, it is no longer acidic. It's not producing it's not acid. It's also not a base anymore. So we've neutralized the basicness of ammonia by making the ammonium ion out of it. So yes, lots of chemical reactions, lots of materials that will bind up hydrogen ions in concentration, and as we get fewer and fewer of these hydrogen ions, and then eventually get no hydrogen ions but just a bunch of chemicals that if a hydrogen ion suddenly appears it would whisk it out of solution, then we become more and more basic. So this is a decrease in H+ ions until we get to, effectively like negative H+ ions, because there's so many of the basic molecules around that a hydrogen ion would not last for even a blink of a second if it were dropped into the solution. Okay, so acids and bases hopefully makes sense, and hopefully you're recalling then the pH scale. So homeostasis in body fluids is, we've got a very narrow little range. It's slightly alkaline, so 7.35 to 7.45. That is homeostasis in body fluids. You can survive if you're anywhere between 6.8 and 8.0. But if you get outside of this survival range, and you may have some consequences of it, you might get crampy if you're becoming too alkaline, or you may get a little dopey if you're becoming too acidic. You can live through that. But if you get much lower than 6.8 or much higher than 8.0, then you're looking at coma on the acidic side, or convulsions and death, eventual death, on the alkaline side. Okay, so there's what homeostasis is. So where then do we come up with, why do we get hydrogen ions in the body? Well, it turns out that most of the hydrogen ions that wind up in our body fluids are produced as a byproduct of the common metabolic processes that we're constantly running. So, if you remember from the respiratory system, you dissolve CO2 in water. Well, that creates carbonic acid, and then carbonic acid falls apart, releasing a hydrogen ion, and then leaving bicarbonate ion behind. When we go about doing respiration of glucose, but we don't have enough oxygen to do it, and we talked a lot about this back when we were talking about the muscular system, So when we do anaerobic respiration of glucose, that's respiratory, or that's cellular respiration, but without, well, not cellular respiration. It's the first step of cellular respiration. When we don't have carbon dioxide, we produce a material eventually called lactic acid. Lactic acid, C3H2O3, will also drop off hydrogen ions. Oh, by the way, while I want you to know carbonic acid, I'm not going to ask you to learn the chemical formula for lactic acid. When we start breaking down fatty acids in order to get to make glucose from them so that we can then use that glucose to make ATP, we frequently yield these structures or these chemicals called ketone bodies. The two that are acidic are acetoacetic acid and beta-hydroxybutyric acid. These two ketone bodies will also drop off hydrogen ions in solution. So let's say you're going on a diet, you're not eating much in the way of carbohydrates, and because of that you're burning fat in order to make glucose so that we can make ATP, and in the process lots of these ketone bodies are going to be produced. If we go about using amino acids that happen to have sulfur in them, and we use those amino acids to try to make glucose so that we can make ATP, then we produce a material called sulfuric acid. That's wrong way. There we go. H2SO4. H2SO4 also will, when it dissolves in water, will drop off a hydrogen ion. And then last of all, as we break down phosphoproteins and nucleic acids, so phosphoproteins are proteins that have phosphorus in them, and nucleic acids also have phosphorus in them. So when we break those down, we can produce phosphoric acid, that's H3PO4, which, no surprise, will also drop off hydrogen ions. So many of the hydrogen ions that accumulate in body fluids are sourced back to metabolic processes that we're running in the body. And if we eat acidic foods, or if we start absorbing before we fully neutralized chyme, then we can definitely get hydrogen ions from the food that we eat as well. So lots and lots of sources of hydrogen ions in the body. Alright, so we get lots of hydrogen ions in. Some of those materials that we were just talking about we describe as strong acids. Others we think of as weak acids. And likewise, there are strong bases and weak bases. So hydrochloric acid is considered a strong base. If we were to take 100 molecules of hydrochloric acid, dissolve them in water, we'd find that, now I'm making this number up, but the point is to get across the idea, maybe 90% of those hydrochloric acid molecules will break apart, releasing the hydrogen ions. So if we put in 100 HCl, we might wind up with, say, 90. And again, I'm making that the actual percentage I'm making up, but it gets the idea across. We might wind up with 90 hydrogen ions running around. On the other hand, if we were to dissolve, say, something like carbonic acid, H2CO3, into our solution, maybe only 30% of those carbonic acid molecules are going to drop off hydrogen ions. So we might only get 30 hydrogen ions that are then in the solution. That's the idea of a strong acid versus a weak acid. Strong acids have way more of their molecules that will break apart and release a hydrogen ion, while weak acids, they tend to have way fewer of their molecules releasing hydrogen ions. So strong acids versus weak acids. Bases, strong versus weak bases, are exactly the same. So Sodium hydroxide is a strong base. If we put that into water, probably 90% or so of them would drop off an OH ion. Whereas something like ammonia might have a much smaller number. It's kind of it's a weaker base, and so maybe only 40% of the ammonia molecules are going to make NH4 plus and absorb or pick up those hydrogen ions that might be running around. So that's the idea of strong acids, weak acids, strong bases, weak bases. All right, so I think we've now got everything defined. So because so many of the metabolic reactions in the body produce acids, acid shifts are much more likely to occur than alkaline shifts. And so maintaining acid-base balance generally involves eliminating hydrogen ions from the body. And we got 3 ways that we can go about eliminating acids. The first is what we're going to talk about in the rest of this lecture. The other 2 we're going to look at in the next lecture. So chemical buffer systems are the first way. It's like the first line of defense against an acid shift in the body. Second line of defense would be respiratory excretion of carbon dioxide. Third line would be the renal excretion of hydrogen ions, so tubular secretion of hydrogen ions. So let's take a look at the 3 different chemical buffer systems that we have in the body. So buffers, one more definition here, buffers are compounds that can stabilize the pH of a solution, and they do it because they can both release hydrogen ions if things are becoming if things are becoming basic, and then so pH is going up, and so as they release hydrogen ions, they cause the pH to decrease again, to get back toward homeostasis. And here's the cool thing about chemical buffer systems, is that not only can they release hydrogen ions, but if things become really acidic, they're also capable of binding hydrogen ions. And as they bind them, they pull them out of solution and effectively neutralize them. And in response, then pH increases. So buffers are capable of doing, of going both ways, of being able to act as a weak acid or act as a weak base, either picking up hydrogen ions or dropping off hydrogen ions. And and these chemical buffer systems that we have are the first line of defense for maintaining homeostasis and pH, because they're found almost everywhere in almost every solution in the body. There's one of these or more of these chemical buffer systems. And because it's a chemical reaction, they tend to happen very rapidly. So they can buffer acids almost immediately upon their production. So there are 3 chemical buffer systems. One of them you already know the chemical reactions to, and I'm going to expect you to be able to talk to me about those chemical reactions. So the carbonic acid, bicarbonate buffer system is the first one. There is the phosphate buffer system. I can't draw a straight line to save my life. And there's the protein buffer system. Excuse me. So we're going to look at all 3 of these. So carbonic acid bicarbonate buffer system, it is present in both the intracellular and extracellular fluid compartments. And I almost think of these equations as being like a balance. So if I can draw like an old-timey balance, right? So we've got a little dish on each side, and you can imagine that on this side of the equation it's H plus HCO3 minus, and on this side it's H2CO3. So if we are seeing an excess in hydrogen, that's going to tip our balance a little bit, because on this side we wind up with more hydrogen ions in our dish, right? Too many hydrogen ions. So the job then of the buffer is to balance things out again. And it balances things out by finding every bicarbonate ion that's around, reacting it with one of the hydrogens, H2 or HCO3 here, minus, react with this one. And so pretty soon, as we use all of the bicarbonate ions that happen to be in solution to bind up the hydrogen ions, then we lighten up this side of the scale, and because we're making more H2CO3, we kind of make the other side of the scale a little heavier. And so we wind up getting back into homeostasis, having it nice and balanced by shoving everything kind of this direction. So that's what I'm showing here with this arrow going this way. If we have an excess of hydrogen ions, then that pushes the chemical reaction toward the carbonic acid side of the equation, then restoring equilibrium, restoring balance, which then is getting us back into homeostasis. If, on the other hand, so if this is like that condition here, under alkaline conditions, the opposite is happening. Under alkaline condition, we're making H2CO3, H2CO3 is out there, and we've got a bunch of let's see how to explain this. I hope that my ideas aren't breaking down now. But under alkaline conditions, we've lost hydrogen ions. So something has ripped away all the hydrogen ions. And so we look around. Where's a source of hydrogen ions that we can move over to this side of the equation? And the source of hydrogen ions that we look to is the carbonic acid that is laying around. And so if there's carbonic acid laying around, all we have to do is rip a hydrogen ion off. And now we have restored, oops, there's no H2 there, it's just HCO3 minus. We have hydrogen ions around, and we have restored our balance again. So if things have become really alkaline, we look for something to give us a bunch of hydrogen ions to make things a little bit more acidic, to help go and interact with all these materials that are the alkalinity. So yeah, here's what we need to help make that make sense. So we've got a bunch of this OH minus laying around. If we can rip the hydrogen off of carbonic acid, free it up, then we can take this OH minus and react it with the hydrogen ion, and we get water. So so now we've neutralized all this stuff over here that was making us basic. That's the piece I was missing. So the more hydrogen ions that we rip off, then the more water we make out of them, and we reduce the amount of alkalinity that there is in the material. And we've reduced or we've shifted a strong base to a weak base. And the weak base is going to tend to be, hanging out over here as the H2CO3, the carbonic acid, rather than as a bicarbonate ion. Okay, hopefully that made some good sense. I'm not sure if it did or not, but the idea is we're trying to keep things in balance so that pH is staying at 7.4 on average, between 7.35 and 7.40, or I'm sorry, 4.5. That's our range that we like. So 7.4, of course, being average. And so if we get too much acid running around, then bicarbonate ion picks it up. If we get too much base running around, then the carbonic acid drops off a hydrogen and we're fixed. Okay, so I want you to learn these reactions. reactions. I will expect you to be able to talk well about that. So the phosphate buffer system, it's the exact same situation. We just have a different ion that we're working with. So here is monohydrogen phosphate. Over here it's dihydrogen phosphate. So we just do the exact same thing that we talked about with the bicarbonate carbonic acid buffer system. With the phosphate buffer system, we're trying to maintain a balance here, and we use this chemical reaction to do that balance for us. So it's the exact same idea, just with different a different chemical that's doing it for us. So that if things get too basic, then we use HPO4. We can rip off that hydrogen, and we can react it with whatever it is that's causing the base, and we convert our strong base into a weak base in the form of this monohydrogen phosphate. If things get too acidic, then we look around for monohydrogen phosphate. We react it with the hydrogen ions that are running around and make more H2PO4, and ta-da, problem resolved. So it's the exact same idea. So phosphate buffer system, it is also found in the intra and extracellular compartments. It's particularly important in controlling hydrogen ions that are produced inside of cells and in urine and developing urine. So we can control pH using this phosphate buffer system. Last of all is the protein buffer system. So this includes the plasma proteins that are found in blood plasma, the albumins especially, and the intracellular proteins that are found in the intracellular compartment, including hemoglobin. So we got hemoglobin there inside those red blood cells, and if you recall, we're busy using we're busy using the bicarbonate ion to carry around the CO2. So there's lots of hydrogen ions that are produced in that process. So the protein buffer system is very important, and proteins are built of amino acids, and amino acids have functional groups on them that can act as buffers. So carboxylic acid is this is carboxylic acid. Let me just draw an amino acid here. So this is an amino acid. It's a carbon that has a carboxylic acid attached at one end, an amino group attached to the other, a hydrogen attached in the third location, and then up here in this location is the variable group. It could be just a hydrogen ion. It could be something really complex like a 6-carbon ring with lots of hydrogens attached to it and double bonds every other, in lots of locations. So there's lots of things that this R can be. There's 20 different structures that might be attached on there. So it could be something really simple, or it may be something very complex. But the carboxylic acid group, it has the capacity of dropping off this hydrogen here, leaving kind of a negative O in its back there, and an H+. So this hydrogen ion can run around then and buffer any alkalinity that might occur. If, on the other hand, there's lots of acidicness, then this nitrogen end, remember how nitrogen doesn't mind bonding, it prefers to make 3 chemical bonds, but just a second ago we were talking about how ammonia can bind up an additional hydrogen ion. So it can also pick up a hydrogen ion there, making this end positive, and so it can grab a hold of, that amino group can grab a hold of extra hydrogen ions, and then later on release it even. So it can bind it, hold it, and if things become alkaline again, then it can drop off that hydrogen ion. And then depending on what the R group has, there may be functional groups there that can also act as buffers. So proteins have a great capacity to also act as chemical buffers in our body. So lots and lots of ways then that we can go about using these chemical buffer systems to help maintain homeostasis in pH by either grabbing up or dropping off the hydrogen ions that are running around. Okay, so there we have it. Everything you ever wanted to know about chemical buffer systems, but were afraid to ask. So you know how to reach me if you have any questions.